Hello, this is
Meg's.
and Michaela's ILV1 retake. In this lab video, we will be demonstrating how to set up a slide on a compound microscope. We will also be explaining and identifying the 5 chordate characteristics in a tadpole, Amicides, adult lamprey, Amphioxus, and by dissection, a sea squirt. Finally, you will learn why these chosen invertebrates were studied in this lab and how they relate to understanding vertebrate evolution. To set up a slide on a compound microscope, it should be plugged in with the stage at the lowest position, and the revolving turret should be clicked into position at the lowest power objective. Now that your compound microscope is ready, we can mount the slide. To hold the slide on the stage, hold open the stage clip. Place the slide on the stage and gently relieve the stage clip so your slide is securely in place. Now, your slide is mounted. What's next? You can move your slide across the stage and into the field of view by turning these dials here. There are 2 knobs along the right side of the microscope. The coarse adjustment can be adjusted until your slide is in broad focus. The fine adjustment knob can be adjusted to achieve precise focus. You can also change the illuminator and diaphragm for light adjustment. From this point, you can change your objective to a higher power if need be, and repeat these steps for each higher objective power until you reach perfect focus.
All chordates possess certain characteristics at some point during their life that may be apparent during transitory embryological development or may persist as a permanent adult structure. I will now identify these 5 characteristics in the following specimens. This is our Ascidian tadpole specimen. They have a well-developed notochord here, which is a longitudinal, stiff, and flexible rod made of cartilage that supports the body. In the Ascidian tadpole, it appears as a series of box-like cells extending from the tail tip to the body. The dorsal hollow nerve cord here, filled with CSF, may be modified into the spinal cord. In this structure, it is dyed dark blue and appears as a tube-like structure dorsal to the notochord, and it also extends from the tail tip to the body. We have the postanal tail, which is apparent here, that extends past the gut region. And we have our pharyngeal slits located in the pharynx region that appear as off-red spots. And this dark red structure over this way is our endostyle, which secretes iodine and mucus that traps food particles and is located along one side of the pharynx.
Now we will be dissecting an adult sea squirt. First, we remove the tunic. This protects the skin so we can clearly see the internal anatomy. First, the incurrent siphon, or the mouth, is here and it intakes food. That then leads into the pharynx, where pharyngeal slits are apparent. Along one side of the pharynx lies the ribbon-like endostyle. The pharynx leads into a short and narrow esophagus, which then leads into the stomach. The stomach leads into the intestine that ends with the terminal anus, and waste leave the body via the excurrent siphon. The notochord and dorsal hollow nerve cord are absent. The nerve cord is modified into a ganglion that extends nerves to the body and is located between the siphons.
This is our Amphioxus specimen. The first structure pointed out here is the notochord, which runs the length of the lancelet and is also located right underneath the dorsal hollow nerve cord, which also runs the length of the lancelet and can be observed right below the dorsal fin ray boxes. Here is our pharynx, which has the pharyngeal slits, and we have the endostyle, which are longitudinal ciliated grooves on the ventral wall. And then finally, we have our postanal tail, which extends past the gut region. This is our Amicides specimen. The pharynx here draws water in across the gill filaments of the gill pouches and out through the external gill slits and pharyngeal slits. The subpharyngeal gland, this dark red one here, is likely homologous with the endostyle of the non-vertebrate chordates, and this lies ventral to the pharynx. We have the spinal cord here, which is modified from the nerve cord. This connects to the brain and lies dorsal to the notochord. And then we have our postanal tail down here with our cracked slide, but this is where our caudal fin would be, the postanal tail. Ammocetes and amphioxus are both visually similar, but here are some internal differences. Amphioxus retains their notochord, while in ammocetes, their notochord modifies into their spinal cord. Ammocetes have a subpharyngeal gland that is homologous with the endostyle that is still present in our amphioxus specimen. Some other prominent internal differences in ammocetes would be the liver, which is here, as well as the gallbladder, and then the heart, which would be here, that we'll display in a second. A little bit of a better photo of the heart right after the pharynx is that clearer structure, and then we have that red lump right after, that is the liver along with the gallbladder. The final differentiation between amphioxus and ammocetes is that the ammocetes show 3 fleshy bumps that encase the cranium. We have the forebrain, the midbrain, and the hindbrain right behind it.
This is our adult lamprey specimen. The notochord here extends from the tail tip to just below the ventral surface of the brain. So it runs all the way through here. In this specimen, the nerve cord evolved into the spinal cord, extending posteriorly from the hindbrain. So that is right above the notochord. Internal and external gill slits are apparent, which are right here. We also have the dorsal and the caudal tail present, and the endostyle that eventually turns into a thyroid gland is here right by the pharynx. Knowing basic invertebrate biology can help us understand vertebrate evolution. It is important to study protochordates because they give us ideas of what the more primitive species are like. They display some or all diagnostic characteristics of a chordate. These characteristics give us an idea of primary vertebrate evolution. We can see a relationship between the most primitive species and more complex species by examining the morphology, embryology, histology, and physiology of functional organs in these chordates. For example, we study lampreys, which retain the 5 chordate characteristics and have a slightly new structure that is modified from the original characteristics. Because we know that Urochordata and Cephalochordata contain all 5 chordate characteristics at some point in their life, we know lamprey and all vertebrates share a heritage to all early chordates. Another example is pharyngeal slits. These are gills in fish and are pouches in humans, which is evidence of ancestry. This precedent allows us to properly assemble a phylogenetic tree to help us determine evolutionary trends between invertebrate primitive species and the more complex vertebrate species, like us. To make sense of the history of development and function of an organ or structure in vertebrates, we look among invertebrate chordates for the possible origin.